settle in and let your breathing slow, as if you are floating just beneath a pond's dark surface. The water is still, the insects are small and busy, and the ripples move like quiet thoughts. You think the lake is empty at night, peaceful and simple. It is peaceful, but it is not empty. Every freshwater fish carries a tiny living neighborhood, so small you could miss it between heartbeats. Tonight, with your mind softening, you will drift with the sleepless scientist through skin, fins, and gills, meeting gentle hitchhikers, patient worms, and the slow logic of life cycles. Nothing here is a jump scare, just a calm ecosystem doing what it has always done, one sleepy routine at a time. You hover near the shoreline, where the water smells like wet leaves and old wood. A fish passes beneath you, a slow shadow with a bright eye that never quite closes. You might think its body is a sealed private thing, but freshwater is a world that touches everything, all the time. Skin is not armor here. Skin is a soft boundary, coated in mucus, like a thin, slippery raincoat the fish makes for itself. And that mucus, inconveniently, is food, not a feast, not a banquet, more like a steady drip of calories for something small, patient, and not at all romantic. Parasite is a dramatic word. It sounds like a monster under the bed. Most of the time, it is closer to a tenant with bad manners. A parasite is simply an organism that lives on or in a host and takes resources. In human language, it is living in someone else's house, eating their cereal and leaving crumbs. It does not need evil. It needs a life cycle, and freshwater is excellent at supplying one. You watch the fish turn, and for a moment, the moonlight catches its scales like dull coins. Along that skin, you imagine tiny things holding on, not with claws and violence, but with the calm certainty of Velcro. Some are so small, you would need a microscope to feel properly outraged, but the fish can feel them. A light irritation, a persistent itch, a sensation like having a hair stuck inside your shirt, except the shirt is your entire body. The quiet water makes it easy to forget how crowded it is. There are drifting larvae in the plankton. There are eggs on plants. There are cysts sleeping in mud, waiting for a mouth to arrive. From above, the pond looks like a smooth black mirror. From inside, it is a busy hallway, and everyone is brushing past everyone else. The fish does not get to opt out of contact. It breathes by pulling water across gills, all day, all night. That means water is constantly sampling the world and delivering whatever it finds. Bits of algae, grains of silt, tiny crustaceans, and sometimes a living speck with a very specific plan. You drift closer to the fish's flank, near the base of a fin. This is where the first small travelers often begin. External parasites, ectoparasites, meaning they stay on the outside. The classics are tiny flatworms and little crustaceans with the personality of a sticker. In freshwater fish, one familiar group is monogeneans. That word sounds like a philosophy club. In reality, they are small flatworms, often living on skin or gills with attachment hooks. A monogenean does not need a multi-stop tour of the food web. Many go from egg to larva to adult on a single host species, efficient like an unpleasant subscription service. Their larvae called Oncomuricidia, are free swimming for a short time. That is a long word for something that is basically a tiny dart, built to find fish. It swims, it bumps into surfaces, it senses chemical cues. Then it chooses a body and it commits. If you could shrink down and stand there, you would see a translucent speck settle onto the fish. It would anchor itself with a little clamp-like structure called an opistaptor. That is another long word for a simple idea. It is a grabby back end. The worm feeds on mucus and surface cells. It may also nibble at blood, depending on the species. Mostly it is an irritant, a stressor, a slow drain. It is not trying to kill the host quickly. If it kills the fish, it loses the house. And parasites, for all their reputation, are often very practical. You watch the fish flick its body once, a quick shiver. That is the fish noticing something. Fish do not have hands. They cannot scratch, so they improvise. They rub against rocks. They scrape along logs. They flash, turning sideways and vibrating, trying to dislodge what clings. If you have ever had an itch you could not reach, you understand the mood. Now drift your attention to another external guest, one with a more visible shape, fish lice. The name is unkind, but fair. These are parasitic crustaceans, often in the genus Argillus. Imagine a tiny, flattened saucer with legs gliding over skin. It has suction cups to attach. It has mouthparts to pierce and feed. When it bites, it can inject enzymes that keep blood flowing. That is handy for the louse and rude for the fish. To you, from far away, the fish still looks calm. But close up, you can picture the skin responding. More mucus, inflammation, a little swelling, a small leak of energy into the problem of being bitten. Most fish in healthy water carry a few of these annoyances and continue their lives. They eat, they avoid predators, they navigate seasons. The parasite is not a headline event. It is background noise. But background noise matters when you live in it every day. If the fish is stressed, if oxygen is low, if the water is warm and crowded, then the balance shifts. A few lice become many. A few worms become a coating. Then the skin, which is supposed to be a barrier, becomes a busy door, and behind that door are bacteria and fungi, always waiting for a scratch. So the fish responds in slow, simple ways. It rests more. It hides in plants. It seeks flowing water, where oxygen is better and irritation feels less suffocating. Sometimes it visits a kind of freshwater cleaning station. The ocean gets the famous cleaner fish. Freshwater has its own quieter arrangements. Small fish may pick at larger fish, eating loose parasites or irritated tissue. Shrimp and some insect larvae will also take advantage of an easy meal. It is not friendship. It is a transaction. The big fish tolerates the little nibbles because the little nibbles remove worse nibbles. Nature loves a bargain, even when everyone involved looks mildly disgusted. You let the fish drift past, and you keep pace beside it, like a slow satellite. Notice how the fins move, gentle fans in the dark. The fish is not fighting the water. It is negotiating with it. And as you watch the fin edge ripple, you remember that many parasites prefer edges, places with flow, places with contact. places where the host cannot easily scrape them off. Some tiny crustaceans wedge at fin bases. Some worms cling near the tail. Some simply ride wherever the currents of the fish's body take them. And then there is the next level of intimacy, because some guests do not stay on the surface. Some prefer the quiet, dark tunnels under the gills. You follow the fish as it angles upward, then down again, passing through a band of slightly colder water. Its gill covers, the opercula, flare and close with steady rhythm. Each movement draws water in, then pushes it across a structure that looks soft and delicate. Because it is. Gills are not like lungs, tucked safely inside. Gills are exposed. They are a breathing forest, combed by water all day long. Inside the gill chamber, you imagine feathery arches lined with filaments. On those filaments are lamellae, thin plates, where oxygen and carbon dioxide trade places. It is a surface made for exchange, which also means it is a surface made for exploitation. Water brings oxygen. Water also brings spores, larvae, and drifting hitchhikers. A parasite that wants easy access to blood and tissue could do worse than move into the gills. Some monogenians specialize here. They clamp onto gill filaments right where the fish cannot scrape with rocks. The flow never stops, so oxygen is always arriving. And the host's blood is always close to the surface. It is like living in a pantry that restocks itself. The fish responds in ways that look like normal behavior until you know what you are seeing. It breathes faster. It flares its gill covers wider. It hangs near the surface where water has more oxygen, especially at night in summer. It lingers where a stream enters the pond, letting moving water do part of the work. Sometimes it becomes sluggish, not because it is lazy, but because it is budgeting. Energy in a fish is not endless. If breathing costs more, then something else gets less. Growth slows, reproduction waits, muscles tire sooner. That is the quiet cost of a small mouth drinking from your breath. Now, imagine a crustacean parasite that takes gills even more seriously. Some copepods, tiny relatives of shrimp, attach to gills and stay there. They can look like small threads or beads tucked between filaments. They hold on with specialized appendages, and they feed on mucus, tissue, and blood. They do not roam. They settle. They become part of the furniture, except the furniture is biting you. If you could hear the fish's body, it would not be silent. There is the soft rush of water over lamellae. There is the pulse of blood in narrow vessels. There is the faint, constant labor of cells maintaining salt balance, because freshwater is always trying to dilute you. And layered on top of that is the tiny disturbance of parasites feeding. It is not dramatic. It is persistent. And persistence is how ecosystems change without making a sound. In heavy infestations, gills can become damaged, filaments thicken, mucus increases, the exchange surface decreases. It is like breathing through a wet cloth. The fish might gasp near the surface, not for theater, but for physics. Oxygen moves by diffusion, and diffusion needs a thin, clean barrier. Parasites and inflammation make that barrier thicker. So, the fish works harder for the same breath. And in a pond where summer nights already run low on oxygen, that extra effort can matter. But most of the time, again, it stays in the realm of mild. A few worms, a few copepods, a fish that still feeds, still swims, still avoids becoming someone else's dinner. Life does not wait for perfect conditions. It operates in compromises. You let the image of the gills fade back into the dark water, and you keep following the fish. You have seen skin and fins, the outer neighborhoods. You have seen the gills, the breathing forest. Now it is easier to imagine what happens when something slips inside the fish itself. The mouth opens just slightly, and the fish takes in a drifting insect larva. To the fish, it is food. To the parasite, if one is hiding there, it is a bus ticket. Internal parasites do not need to cling to the outside world. They aim for the slow, warm hotel inside. The fish's gut is dark, warm compared to the pond, and full of broken down nutrients. There is movement, peristalsis, squeezing food along. There are digestive enzymes, chemical scissors. There are immune cells, always watching. It sounds hostile, but many parasites are built for exactly this hostility. They do not survive despite the gut. They survive because the gut is predictable. One common kind of internal tenant is a tapeworm, a cestode. Tapeworm is a blunt word for a strange animal. It is flat, ribbon-like, and segmented. It anchors with a scolex, a head that is less a head and more a holdfast. Some have suckers, some have hooks. Then the body grows behind it, a chain of segments called proglottids. Each segment is like a little package of reproductive organs. If you are impressed, lower your standards. The tapeworm's body plan is basically a factory line for making eggs. It does not need a complex brain. It does not need eyes. It lives in darkness, bathing in nutrients. It absorbs food through its surface. It is a creature that outsourced the act of eating to the host. Congratulations, you have become someone else's digestive system. Tapeworm infections in fish can be subtle. A fish may carry one or several and still appear normal. Sometimes there is weight loss, slower growth, less energy. Sometimes nothing obvious happens until the burden is high. And sometimes the damage is not from the adult at all, but from the larval stages that lodge in organs or muscle. Another internal guest is the roundworm, a nematode. These are more like threads, cylindrical, and often surprisingly tough. Some live in the gut, some migrate through tissues, some encyst, forming a little capsule in muscle or body cavity, waiting. Waiting is a theme here. The pond runs on waiting. A nematode larva might sit in a cyst for months, doing very little. Not because it is lazy, because it is between chapters. It is in a stage designed for survival, not for growth. And survival stages are often stubborn. They resist digestion. They tolerate cold. They endure low oxygen. They wait out winter like a secret. If you want a calmer way to think about internal parasites, think about storage. The parasite is stored in the fish until the right predator comes along. Because many internal parasites do not finish their lives in fish. Fish are often the middle of the story, a temporary address, a stepping stone in a food web that moves energy and also moves passengers. The fish eats a tiny crustacean. The crustacean contains a parasite larva. The parasite enters the fish. Now the parasite arrives at its final home, where it can become an adult and reproduce. It is not personal. It is logistics. And logistics is one of the quiet engines of nature. You might wonder how the parasite survives the fish's defenses. Fish have immune systems, of course. They have white blood cells, antibodies, inflammation. They can recognize foreign proteins and attack. Parasites respond with disguise and endurance. Some coat themselves in molecules that look like the host. Some hide inside cysts. Some live in places with weaker immune surveillance. Some simply reproduce faster than the host can clear them. It is not a heroic battle. It is a slow negotiation, carried out in chemistry. And because it is slow, it is often invisible. The fish goes on being a fish. It hunts. It avoids shadows from above. It rests among plants. Meanwhile, inside, something is quietly turning host nutrients into parasite eggs. Of course, getting inside usually requires a detour because parasites love complicated journeys. So you zoom out just slightly from the fish's body and you look at the pond as a map, not a scenic map, a transit map. In this map, every mouth is a station and every stomach is a transfer point. You do not travel by choice. You travel because you were swallowed, and your entire body plan is built around that insult. You begin, often, as an egg, not a pretty egg in a nest, a microscopic capsule sealed like a cheap plastic container designed to survive disappointment. The pond receives these eggs the way it receives everything else, quietly, repeatedly, with no ceremony. Some eggs sink into mud and wait. Some cling to plant stems, where snails will graze like sleepy vacuum cleaners. Some drift in the open water, bumped around by currents, as if the lake is slowly shuffling a deck. And you, the egg, are the card someone is meant to draw. This is why parasite life cycles feel complicated. It is not because parasites love complexity for its own sake. It is because the pond is a chaotic place and probability is cruel. If you only had one host to find, you would probably fail. So you hedge your bets. You split your life into stages, like changing outfits for different rooms. One stage is for surviving outside. One stage is for finding a snail. One stage is for enduring a fish. One stage is for adulthood, reproduction, and the final rude act of leaving eggs behind. Forget what you learned in school, that nature is efficient in a clean, elegant way. Nature is efficient the way a junk drawer is efficient. Everything is in there, and somehow it works, but you do not want to touch it. You follow a drifting egg down toward the shallows. The water here is warmer by a whisper. It smells like algae and wet stone. A snail moves along a reed, slow enough to feel like a decision. Snails are gentle machines. They scrape surfaces for biofilm, swallowing whatever is glued to life. And that is the point. Many parasites do not chase their hosts. They let hosts do the work by being edible at the wrong time. A classic group here is the trematodes, also called flukes. They are flatworms, shaped like tiny leaves, and they love a multi-stop itinerary. Their adult forms often live in birds, or mammals, or sometimes larger fish, but their early chapters begin in snails, because snails are everywhere, and snails are patient. If you are a trematode egg, the goal is simple: get eaten by a snail. Inside the snail, the egg hatches into a larva called a mericidium. It is small, ciliated, and frantic by parasite standards. It swims like a dust mote that learned panic. But that panic is short-lived because it quickly becomes something else. Inside the snail, it transforms into a sporocyst. That word sounds like a space station. In human terms, it is a living sac, and it does something unsettlingly practical. It clones itself. Not metaphorically, it turns the snail into a factory for making more parasite larvae. You thought parasites were just thieves. Sometimes they are also managers. The snail does not explode. It does not scream. It continues gliding along, feeding, leaving a faint trail. But inside, resources are being redirected. Energy that could have made snail eggs becomes parasite bodies instead. It is a quiet rerouting, like a budget cut you only notice months later. Eventually, the snail releases another larval stage called a cercaria. These are the ones that look like tiny tadpoles, built for escape and travel. They leave the snail in little bursts, often timed to light and temperature. Morning, afternoon, warm seasons, the hours when fish are active. Not because the parasite watches a clock, because natural selection built a schedule into its chemistry. You float beside a cercaria as it enters open water. It wiggles, searching for a host. Some cercariae directly penetrate fish skin or gills. Others find insect larvae. Others aim for amphibians. But tonight, you are following the ones that want freshwater fish. They find a fish by bumping into it and by sensing faint chemical trails, mucus, amino acids, the soft signature of a living body. Then they do something bold. They burrow in. The fish feels it as a tiny sting, if it feels it at all. The cercaria sheds its tail the way you might drop a backpack at the door. Then it encesists, forming a metacercaria, a sleeping stage wrapped in a protective capsule, tucked into muscle, or under skin, or in the eye. Yes, sometimes the eye. Congratulations, your vision is now part of somebody else's long-term plan. This is where the pond's transit system becomes truly sleepy, because a metacercaria can wait weeks, months, sometimes longer, depending on temperature and host metabolism. The fish continues its routine. It feeds, it hides, it drifts. It carries the cyst like a quiet pebble under the skin. The parasite is not in a hurry. A parasite that rushes is a parasite that dies. Now, you follow the next transfer. A heron stands at the shoreline like a patient statue. It does not need to know anything about trematode biology. It only needs hunger. The heron strikes and the fish becomes food, and the cyst enters a new digestive tract. Inside the bird, the cyst opens. The fluke matures. It attaches in the gut, or the bile ducts depending on the species. It feeds, it mates, it releases eggs. Those eggs leave the bird with the same lack of drama that started the story. A drop into water, a speck into mud, a new loop. When you hear life cycle described in a textbook, it can sound like a diagram, arrow to arrow, stage to stage, neat and confident. In the pond, it is not neat. It is a probability cloud. Most eggs fail. Most larvae do not find a host. Most cysts never reach the right predator. That is why parasites produce so many offspring. They are not being extravagant. They are compensating for reality. And reality is that the pond is full of mouths, and most of them are the wrong mouths. Speaking of wrong mouths, you can follow another route that uses an even smaller vehicle. Tiny crustaceans, the drifting snacks of freshwater. Copepods and water fleas, zooplankton that pulse through the water like living commas. They filter feed, they graze. They are the base of many fish diets, which makes them perfect from a parasite's point of view for moving up the food chain. Consider a tapeworm again, but start earlier. Many fish tapeworms begin in a tiny crustacean. The egg hatches into a free-swimming larva, a Curiosidium, covered in cilia. It looks like a glittering sphere under a microscope. It gets eaten by a copepod. Inside, it becomes a procercoid, a larval form, built to survive and wait. Then, a small fish eats the copepod. Now the parasite becomes a plerocercoid, larger, more developed, and ready for the next step. Sometimes that small fish is already the final host. Often it is not. Often, the small fish is just a hallway you pass through. A larger predatory fish eats the small fish. The parasite moves again, deeper into the chain, toward a body big enough to support an adult. And somewhere along the way, you see the logic. Not moral logic, mechanical logic. Each host is chosen not for kindness, but for likelihood. Snails are likely to eat eggs. Copepods are likely to eat larvae. Fish are likely to eat fish. The pond is not a peaceful painting. It is a machine made of appetites. But it is a slow machine. It creaks gently. It turns without anyone noticing. If you want to feel how quietly this works, imagine you are the parasite larva inside a copepod. The copepod's body is transparent, and the world is a warped lens of moonlight. Its gut pulses as it feeds. Its legs beat in short, jerky strokes, like a swimmer who never learned to relax. You sit inside, small and folded, while the copepod dodges shadows. A fish approaches. The copepod tries to flee. It cannot flee fast enough. The fish inhales, and in a single pressure wave, your entire universe becomes throat. You have no opinions about this. You are not angry. You are adapted. Your outer layers resist digestion just long enough. You wriggle free into the fish's gut, and the gut becomes your new climate. Warm, dark, rhythmic. You anchor or you bore into tissue, depending on what you are. And then you wait again, because patience is the one trait every successful parasite shares. There is a softer way to think about all of this. The pond is full of travelers that do not have legs, so they borrow legs. They borrow snails. They borrow insects. They borrow fish. They borrow birds. Every host is a borrowed vehicle, and every vehicle eventually breaks down. That is not cynicism. That is ecology. But if you are the fish, none of this feels like a grand system. It feels like mild discomfort and occasional fatigue, in the sense that you cannot quite grow as fast as you should. It feels like having a budget you did not agree to. Now drift your attention toward the littoral zone, the shallow edge where plants grow thick. This is where many intermediate hosts live and where the parasite transit system gets crowded. Snails cling to stems. Insect larvae hang beneath leaves. Small fish pick through debris. And every surface is coated in biofilm, the living slime that feeds half the pond. Biofilm is not glamorous. It is bacteria, algae, fungi, tiny protozoa, all stuck together in a gel. It is the pond's shared pantry. And because it is shared, it is also a shared risk. Eggs stick to it. Larvae hide in it. Cysts settle near it. If you are a fish that grazes plants or nibbles algae, you are also grazing on invisible possibilities. Some parasites do not even need you to eat a host. They only need you to brush past. A Cercaria can penetrate your skin in seconds. It releases enzymes that soften the surface and it pushes through. A tiny wedge. The fish's immune system reacts, but often too late, to stop the first entry. Then the parasite seals itself in a cyst, and your body walls it off. This is one of the strangest compromises in biology. Your body cannot always remove the parasite, so it builds a room around it, a capsule of tissue, a little prison. But the prisoner wanted a safe room anyway. So both sides call it a solution, and both sides are slightly wrong. There is a famous example that freshwater anglers sometimes notice as little black dots on fish skin, black spot disease. It is not a single disease, but a sign of trematode metacercariae encysted under the skin. The black color is the fish's pigment cells responding, darkening around the cyst. From far away, the fish looks peppered. From the parasite's view, it is wearing camouflage made of fish panic. The fish usually survives. It swims with its dotted pattern, but the dots may make it more visible to predators, depending on the setting. And that can help the parasite, because the parasite needs the fish to be eaten by the right final host. This is where the story becomes quietly unsettling. Some parasites do not just ride the transit system, they nudge it, not with intelligence, with chemistry, with small changes in host behavior that tilt probability. Sometimes infected fish swim closer to the surface. Sometimes they become a bit slower. Sometimes they flash more often, drawing attention. Even a tiny shift can matter when predation is a numbers game, and if you are trying to reach a bird's gut, being eaten is not a failure. It is graduation. That is the pond's idea of a diploma. You watch a small fish hover near the reeds. It makes short, nervous starts. Its movements are not smooth. This could be many things. Low oxygen, a near miss with a predator, a bit of irritation from gill parasites, or it could be the subtle drag of larvae encysted in muscle. Most of the time, no single cause is dramatic. It is the stack of small burdens that changes a life, which is also, inconveniently, true for you. Speaking of small burdens, consider leeches. Leeches are not always parasites, but many are. They are annelids, segmented worms, relatives of earthworms, built for aquatic patience. A leech does not chase like a shark. It waits like a sock lost in laundry. You find one on the underside of a rock. It is flattened, dark, and glossy. A small strip of living rubber. It senses vibration and warmth. A fish passes close. The leech extends, attaches with a sucker, and begins to feed. It releases anticoagulants, so blood flows. This is why leech bites can ooze longer than you expect. In fish, the feeding is often brief, a sip, a swelling, then detachment. The leech drifts away like a leaf, full and slow. It is not interested in killing its host. It wants a meal, then time to digest, then another meal. Some leeches also act as vectors, carrying blood parasites from one fish to another. In that sense, the leech is not just a parasite, it is a syringe with opinions. But again, no malice is needed, only survival. Now, stay near the plants, because plants are where the pond keeps its small, long schedules. Parasites often synchronize with seasons. Warmer water speeds metabolism. Snails reproduce more, fish feed more, birds migrate and return. So parasite transmission often peaks in late spring and summer, when bodies move and mouths stay busy. In winter, the pond slows. Fish drop into deeper water, conserving energy. Snails become sluggish. Larvae survive in cysts, in eggs, in mud. It is not inactivity. It is storage again. Life packed away, waiting for better conditions. Some parasite eggs are remarkably tough. They resist drying. They tolerate freezing. They survive passage through a gut. If that sounds unfair, it is. But the pond is not built for fairness. It is built for persistence. And parasites are specialists in persistence. You can watch this persistence play out in a single snail. The snail is a spiral of dull amber. It grazes along a stem, and its tentacles taste the water. Inside, trematode stages occupy space, redirect nutrients, and sometimes even sterilize the snail. Parasitic castration is the blunt term. The snail stops investing in its own reproduction because the parasite has taken over that budget. The snail becomes, in a way, an extension of the parasite's body, a living nursery. This sounds horrifying when you say it fast. When you say it slowly, it is just biology being indifferent. The snail does not mourn its lost future. It continues to feed, to move, to exist. Nature rarely gives you the dignity of understanding what happened to you. And if you are listening in bed, safe and warm, you can appreciate that ignorance is sometimes a gift. Now follow a different passenger. Not a fluke, not a tapeworm, but a protozoan. Single-celled organisms can parasitize fish too, often on skin and gills. One example is Ichthyophthirius multifilius, usually called ick. That name is mercifully short because the experience is not. Ick is a ciliate, a tiny cell covered in hair-like structures for movement. It burrows under the fish's skin, or gill epithelium, feeding and growing. The fish develops white spots, like grains of salt. Each spot is a parasite under the surface, protected for a while from treatments and from the immune system. Then it drops off, sinks, and divides. One parasite can become hundreds, then thousands, each one ready to seek a new host. This is why itch outbreaks can explode in aquariums. In the wild, the pond's volume and complexity dilute the effect. But stress can change that. Warm water speeds the parasite's cycle. Crowding increases contact. Low oxygen taxes the fish. And suddenly, the quiet balance tilts into a visible problem. You see the pattern forming now. The pond tolerates parasites the way a body tolerates scars. A few are normal. Many become trouble, especially when the environment is already strained. Which brings you back to the idea that parasites are not separate from ecology. They are part of it, stitched into food webs, seasons, and behavior. They are one of the ways populations are kept from becoming too perfect. A pond without parasites would be like a town without weather. It sounds pleasant until you realize the weather is part of what keeps everything moving. Speaking of moving, return to the drifting zooplankton. Watch a water flea, a Daphnia, pulse through the water like a beating comma. Daphnia are not fish, but they are part of fish parasite stories. They can carry larval stages of certain worms, and they can also host their own parasites. The pond is layered like that. Parasites inside prey, inside predators, inside water. A nesting doll made of appetites. If you are a fish-eating Daphnia, you are also sampling a microbial world. Bacteria and viruses move through water too. Some viruses infect fish, causing lesions, bleeding, immune suppression. Often the difference between a mild parasite load and a deadly outbreak is not the parasite itself. It is the background conditions, temperature, pollution, nutrient runoff that fuels algae blooms, which later crash and consume oxygen. Crowding in small bays when water levels drop, stress hormones in fish that change immune function. Cortisol is a common one, a stress hormone found in many vertebrates. In fish, chronic stress can reduce immune responses, making parasites more successful. So the parasite story is never just about the parasite. It is about the pond as an environment that either supports resilience or quietly removes it. Now, let your attention drift toward a slow, gelatinous ribbon attached to a fish's gill filament. This is where you meet some copepod parasites in more detail, because they deserve the discomfort. Copepods are tiny crustaceans, and most of them are harmless little commas in the water. But a few species discover the joy of real estate, specifically gill real estate. You watch one anchored there, half hidden in the breathing forest. Up close, it is less like an insect and more like a living clip. The parts you would call arms are modified into hooks and clamps. The body is soft in places, armored in others, like a suitcase built for rough travel. It does not need to swim much anymore. It has already arrived. Some parasitic copepods have a free-living youth, a brief searching phase. They drift as larvae, sensing currents, tasting chemistry, bumping into the world. Then they find a fish, and their body changes direction in life. Attachment becomes the whole career. On gills, they feed on mucus and tissue, sometimes drawing blood. The gill filaments respond by thickening, the way skin makes a callus. That sounds helpful. Until you remember, gills need to be thin. A thick gill is a gill that bargains away oxygen. You imagine the fish during a warm night, when oxygen is already low. Its muscles want energy, but its gills are negotiating with unwanted tenants. So it hangs near the inflow, where water moves and tastes fresh. It looks like rest. It is actually triage. There are parasitic copepods with names that sound almost polite. Ergocylus is one of them, often called gill maggot in fish-keeping circles. The adult female clamps onto the gill and stays put. The male, in some species, lives more freely and dies sooner. It is an arrangement that feels unfair, which means it is perfectly natural. The female feeds, swells with eggs, releases them into the water. The eggs hatch into knobbly, tiny larvae that look like grainy dots. Those larvae molt into copepodids, more mobile stages, built to seek a host. Then the cycle repeats, not angrily, just reliably. And if you are the fish, you never get a break from the math, because every breath is another sampling of the pond. Every sampling is another chance for a larva to touch you. You think of parasites as rare events, but the water makes contact constant. Even when the fish is alone, it is never alone. Now, move your attention from the gills to the broader shoreline community again, because the gills are just one neighborhood in a much larger housing market. And the shoreline is where so many deals are made. Snails are still grazing, slow and indifferent. Insect larvae are still hanging under leaves, waiting out their own metamorphoses. And somewhere in this soft, plant-shadowed world, parasites are using other helpers. Not always the dramatic kind. Sometimes the helper is just an animal that is easy to eat. Leeches, for example, do more than sip. Some leeches carry protozoans in their gut and deliver them to fish. It is less like a curse and more like poor hygiene. A fish gets bitten, a tiny wound opens, and cells have a doorway. The parasite does not need a clever trick. It needs access. You follow a leech as it detaches and drifts. Its body undulates slowly, like ribbon in a lazy current. It finds another rock, another place to wait. The pond is full of waiting places. And this is why parasites can feel so common. Once you learn to look, the entire ecosystem is built from repeated contacts, grazing, biting, filtering, swallowing. Every action is also an opportunity for transfer. Now, let the water carry you towards something even calmer. A snail nursery, hidden under a raft of duckweed. The duckweed roots hang down like soft thread. They collect debris, bacteria, tiny algae, and yes, parasite eggs. A snail climbs up and rasps at those roots, feeding. If trematode eggs are present, this is where they enter the snail. Inside the snail, the parasite does not stay one parasite for long. It becomes a colony of itself, a branching set of stages. Sporocysts and rediae, depending on the species, keep making more larvae. The snail becomes a living incubator, often for months. It is not a sudden takeover. It is a quiet redirection of calories. The snail still moves, still feeds, still survives. But it may stop reproducing because the parasite has claimed the budget. Parasitic castration sounds like a horror story. In practice, it is an energy strategy. If the parasite can keep the snail alive and feeding, it gets more time. More time means more cercariae released into the pond. And those cercariae are the pond's drifting confetti. Not festive, just numerous. They leave the snail in pulses, often at times when fish are active. Light and temperature act like invisible clocks. A warm afternoon can trigger a release. So can the first bright hours of morning. The cercariae swim with tails, pushing through water like tiny commas in motion. They are fragile in a way that adults are not. They do not live long without finding the next host. So they spend their short time doing one job: contact. They bump into fish, they latch, they probe skin and gills for a way in. Some penetrate and insist as metacercariae, and the fish becomes storage. Some fail and die, becoming protein in the water, recycled without ceremony. That recycling is another quiet truth. The pond wastes very little. Even failed parasites feed something. Now, keep drifting, because there is another helper you have not fully met yet. Insect larvae. The pond is full of them, and many fish love to eat them. Mosquito larvae wriggle near the surface, breathing through little tubes. Mayfly nymphs cling to stems, grazing on algae. Dragonfly nymphs sit like predators in miniature, waiting to ambush. All of them are potential vehicles for parasite stages. Some parasites use insects the way commuters use trains. A larva gets inside an insect, survives the insect's metamorphosis, then waits. When a fish eats the insect, the parasite changes address again. And yes, sometimes the parasite survives, the insect becoming an adult. It rides the transformation like luggage. Then, the adult insect is eaten by a fish, and the luggage finally arrives. If you find that unsettling, that is reasonable. But in the pond, metamorphosis is just another hallway. Now, consider a parasite group that is especially fond of these food web staircases. Acanthocephalans, the thorny-headed worms. The name is honest if you know the pieces. They have a proboscis armed with hooks, like a tiny medieval weapon. It retracts and extends, anchoring into the gut wall of a host. They often begin life in crustaceans or insect larvae. A small aquatic arthropod eats the egg. Inside, the parasite becomes an acanthella, then a cystacanth, a waiting stage, then a fish eats the arthropod. Inside the fish, the worm attaches in the intestine and grows. Some species mature in fish. Others still want the fish to be eaten by a bird. The point is always the same. Move upward, find a gut that fits adulthood, and along the way, the parasite may adjust the behavior of the intermediate host. Infected amphipods, small shrimp-like crustaceans, can become bolder. They swim closer to light, closer to open water, closer to being eaten. It is not a conscious choice. It is chemistry changing risk. The pond does not need mind control in a dramatic sense. It only needs a small tilt in probability. That is enough to change who becomes lunch. You can feel how calm that is. In its own way, the parasite does not need to chase or fight or roar. It needs to wait and nudge. Freshwater ecology is full of nudges. Now, speaking of nudges, you return to the fish again. This time, you watch the fish's behavior with new suspicion. It swims a bit higher than the others. It lingers where the moonlight reaches down, faint and silver. Maybe it is feeding. Maybe it is warmer water. Or maybe it is carrying a few passengers that would benefit from visibility. There are trematodes that insist in eyes, forming cataracts in fish. A cloudy lens makes the fish slower to react. That slow reaction makes it easier for a bird to catch. And for the parasite, the bird is the final address. This is not evil. This is selective pressure writing a simple script. If the parasite's genes get passed on more often when fish are caught, then fish catching becomes part of the design. Over many generations, the pond becomes full of these quiet designs. You might think the fish would evolve to prevent all of this. It tries. But parasites evolve too, and often faster. Short generation times mean more experiments. More experiments mean more successful tricks. So the fish ends up with defenses that reduce harm, not eliminate it. More mucus, better immune recognition, behavioral habits like rubbing and flashing. And the parasite counters with timing, disguise, and sheer numbers. The pond becomes a long negotiation with no final treaty. Now let the scene widen. because not all helpers are small. Some helpers have feathers. You notice a gull on a low branch, half asleep, head tucked. You notice the heron again, patient as ever. You notice in the distance the faint silhouette of a mink near the bank. Predators are part of parasite life cycles more often than you would like, because predators do what parasites need most: move bodies across the food web. When a predator eats a fish, it is not just calories moving, it is parasites changing apartments. Some parasites are not fully grown until they reach a bird. They remain larvae in fish, encysted and quiet. Then the fish is eaten. The cyst opens in the predator's gut, and adulthood begins. Adult trematodes in birds often live in the intestine or the bile ducts. They attach with suckers, feed, and release eggs. Those eggs leave the bird with droppings, returning to water. The lake accepts them the way it accepts everything else, without comment. Even mammals can serve as final hosts. Otters, for example, eat fish and can host adult parasites. So can raccoons, and sometimes humans, if the fish is eaten raw or undercooked. That last part is not meant to scare you. It is just a reminder that food webs do not care who you are. If you put yourself in the wrong place in the web uses you. Now, keep the mood gentle by returning to the simplest version of this. A small fish eats a snail or a crustacean and gains a larva. A bigger fish eats the small fish and gains the larva. A bird eats the bigger fish and the larva becomes an adult. Eggs return to water and the circle closes. It is a bedtime story written in digestion, and it is repeating in thousands of ponds at this exact moment. You might wonder what the fish gets out of this arrangement. Nothing usually. Sometimes a parasite can have minor, weird side effects that are not immediately deadly, but in general, parasitism is not mutualism. It is not cooperation. It is one organism extracting resources from another while trying not to wreck the equipment. And the fish, if it survives long enough, reproduces anyway. That is the compromise. The fish does not need to be parasite-free. It needs to be parasite-tolerant. That is how many wild animals live, not in perfect health, but in workable health. Now, to understand how predators help parasites, you watch a particular moment. A small perch, maybe, darts through weeds with something wrong in its rhythm. A fraction slower, a fraction less cautious. A pike waits in the shadows like a long, cold thought. The strike is fast, and then it is over. You do not hear screaming. Fish do not give you that courtesy. The pike swallows, and inside that swallow, several parasite plans move forward. A larval tapeworm in the perch now sits in a larger gut. A cyst in muscle now meets digestive juices and opens. A roundworm larva in the body cavity is now in a new body cavity. Not every parasite benefits from this move. Some get digested and die, but enough survive that the strategy remains. And if the next predator is a bird, then the parasite's story may finally reach adulthood. There are fish parasites that are practically built around this. Diphyllobothrium, a group of broad fish tapeworms, often uses crustaceans and fish before reaching mammals or birds. Eggs hatch into Curiosities in water. Copepods eat them, becoming the first intermediate host. Fish eat the copepods, becoming the second host. Then a mammal or bird eats the fish, becoming the final host. The adult tapeworm produces eggs, and the cycle continues. If humans are involved, it is usually through raw fish dishes. Again, not a scare, just ecology crossing paths with cuisine. In the wild, the final hosts are often bears, dogs, foxes, seals, birds, any fish eater with the right gut environment. The parasite does not care about names. It cares about temperature, enzymes, and time. And speaking of time, parasites often synchronize with migrations. When certain fish move into shallow water to spawn, parasites benefit from crowding. When birds arrive seasonally, parasites benefit from new final hosts. When water warms, larvae develop faster and transmission peaks. So the parasite calendar is an ecosystem calendar. Even without a brain, a parasite can be seasonal. It is built into its development rates. A cold spring can slow the whole cycle down. A long, warm autumn can extend it. The pond keeps records in the form of who gets infected and when. Now, zoom out further, because sometimes predators are not just fish and birds. Sometimes the predator is a dragonfly nymph eating an infected crustacean. Sometimes the predator is a frog eating an infected insect. Parasites can move through these side routes too. Not always successfully, but often enough to matter. The pond is full of wrong mouths, but wrong mouths still move things around. Even a failed transfer can leave eggs in a new place. Even a dead host can release cysts into sediment. The system is messy and stubborn. Now, you let the predator scene fade into the background. Because after all these transfers, it is useful to ask a simpler question. What does all this do to the lake overall? From far away, parasites look like misery. From farther away, they look like regulation. Parasites rarely wipe out healthy populations in stable environments. Instead, they shave off the easiest victims: the stressed fish, the injured fish, the fish living at the edge of tolerable oxygen. That sounds cruel, and it is, but it is also a kind of ecological trimming, like wind pruning a tree. Parasites can prevent any one species from becoming too dominant. They push energy into different channels. They create selective pressure for stronger immune systems and better behaviors. They also, inconveniently, help keep food webs diverse. If one fish species explodes in number, its parasite load often rises too. That increased load can reduce growth and survival, slowing the boom. Not because the pond cares about balance, because feedback loops happen whether you want them or not. This is why a lake that is too clean can be unstable. Not clean as in clear water, but clean as in simplified. If you remove intermediate hosts, or remove predators, or change shoreline habitat, you change parasite life cycles. Sometimes that reduces disease, yes. Sometimes it creates new problems because the system was held in place by many small frictions. Now consider stress again, because stress is the lever that turns mild infections into outbreaks. When fish are crowded, parasites spread more easily. When oxygen is low, fish breathe harder, pumping more water over gills. More water means more parasite contact. When pollutants irritate gills and skin, the barrier is weaker. When temperature rises, many parasites develop faster. So human changes to freshwater habitats can intensify parasite problems. Nutrient runoff can cause algae blooms, which later die and consume oxygen. Low oxygen stresses fish, and stressed fish become easier hosts. Warm, shallow water in summer can become a parasite accelerator. Not because parasites like you personally, because they like physics and chemistry that favor them. In aquaculture, where fish are raised in dense groups, this becomes obvious. External parasites like fish lice and monogenins can spread quickly. Itch can explode through a tank if conditions are right. Farmers use treatments, filtration, and management to keep loads down. In the wild, the pond uses dilution and complexity. Big water, many hiding places, many species, and fewer constant contacts. Still, even wild fish can suffer during heat waves or droughts. When water levels drop, fish crowd into smaller volumes. Parasite transmission rises, and oxygen drops, a double inconvenience. You can picture it as a slow tightening of the room. Not a sudden disaster, just less space, less air, more contact. Now, because this is a story about freshwater fish and more, you widen your view beyond fish. Amphibians share these waters, and they carry their own parasites too. Trematodes can deform frog limbs when larvae encyst near developing tissues. That is a famous example of parasites meeting pollution and habitat change. More nutrients can increase snail populations. More snails can increase trematode transmission. More trematodes can mean more deformities in frogs. A chain of calm steps leading to a visible outcome. No villain, just cause and effect. The pond is full of these chains. Now come back to fish and think about what a healthy fish actually looks like in nature. It is not spotless. It has scars, minor infections, small parasite loads. It carries a few hitchhikers, the way you carry a few annoyances in your day. Most of the time, it still eats and grows. It still escapes predators. It still spawns when the season arrives. Parasites are part of the normal background. The trouble starts when background becomes foreground. When a fish is already stressed, parasites can tip it over. When gills are already irritated, a few more copepods matter. When food is scarce, a tapeworm's slow drain matters more. When temperature is high, ick divides faster, and the fish loses the race. So the lake's balance is not about eliminating parasites. It is about keeping the background low enough that a fish can still be a fish. It is about friction, not perfection. In a stable pond, parasites act like weather that never makes the news, annoying up close, shaping everything from far away. You watch the water and you realize stability is not the same as cleanliness. Stability is having enough plants, enough oxygen, enough hiding places, enough variety. A pond with many species is harder for any single parasite to dominate because parasites are picky in the most practical way. Many can only use one or two host species well. If those hosts are rare, the parasite spends its life losing the lottery. But if those hosts are crowded together, the parasite wins too often. That is one reason overcrowding is such a problem in fish ponds and tanks. It is not that parasites become evil in captivity. It is that probability stops being shy. When fish are packed close, contact rates rise. When contact rates rise, every free-swimming stage has an easier job. A monogenian larva does not need to search a whole lake. It only needs to drift a few fin lengths. A ciliate, like itch, does not need a long journey. It only needs the next body in line. The fish cannot spread out, cannot hide, cannot dilute the problem. Dilution is one of nature's favorite medicines, not because it cures anything, but because it slows everything down. And slowing down is often enough for an immune system to catch up. Now picture a pond in late summer when the days have been hot. The surface looks calm, but the water is tired. Plants and algae produce oxygen in daylight. At night, they switch to respiration and they consume oxygen instead. So the early morning hours can be the thinnest air a fish will ever breathe. You hover in that pre-dawn water, and it feels subtly heavy. Not to you, perhaps, but to a gill that already has extra mucus, to a fish already spending extra energy on irritation and repair. This is when a mild parasite load matters more, not because the parasite suddenly changes, because the environment stops offering slack. A healthy pond has slack built in. Cooler refuges, flowing inflows, shaded edges, deep water. But when water is shallow, warm and still, the margin disappears. And once the margin is gone, small problems stack up fast. You might think the fish's immune system would simply solve it. It tries, but it is not free. Every immune response costs calories. Inflammation, mucus production, cell turnover, all of it is paid for in energy. A fish that spends more energy on defense spends less on growth, less on reproduction, less on escaping predators. So, parasites can shape populations quietly, not by killing outright, but by changing budgets. In ecology, budget changes are destiny. Now, consider the pond from a different angle. Imagine a pond that has been simplified by human hands. The shoreline plants are removed to make a neat bank. The snails lose habitat, and some parasites lose a key stop. But insects also change, and fish feeding patterns change. Predatory fish might be removed or introduced, shifting who eats whom, and the parasite community shifts with it. Sometimes fewer parasites, yes. Sometimes a different parasite becomes dominant because competitors are gone. The result is rarely a simple improvement. It is usually a rearrangement of costs. Nature loves rearranging costs, and freshwater ecosystems are especially sensitive because they are small. A lake can buffer change with volume. A pond feels every change like a bruise. Now you let your focus narrow again to one ordinary fish. Not a heroic fish, not a doomed fish, just a fish with a normal life. It carries a few monogenians on its gills, a couple of encysted trematode larvae in muscle, maybe being roundworm tucked in the gut, doing its quiet accounting. And it still eats. It still responds to shadows. It still finds a mate when spring returns. This is the reality most wild animals live in. Not disease-free, just functional, which is why parasites can be part of ecosystem health in a twisted way. A complete absence of parasites can mean the system is too sterile, too simplified, or it can mean the hosts are gone, which is a more dramatic kind of cleanliness. So parasitologists often do not ask, are there parasites? They ask, which parasites? And how many? And what is the context? Because context is everything. A few black spot cysts on a fish in a clear pond is one story. A heavy itch outbreak in warm, crowded water is another story. Now let the pond offer you a quieter thought. Parasites tend to remove the individuals already closest to failing, the very young, the injured, the stressed, the ones with poor access to food. This sounds brutal, and it is, but it also means parasites can act as a selective pressure for resilience. Fish with stronger immune responses survive more often. Fish with better behaviors, like choosing oxygen-rich areas, survive more often. Over time, this shapes the population. The pond becomes full of survivors that can tolerate the pond's normal inconveniences. That includes parasites. But when the pond changes faster than evolution can respond, problems appear. A sudden heat wave, a chemical spill, a long drought. Those changes do not give fish generations to adapt. They give fish days. And parasites, always ready to exploit weakened hosts, become more visible. This is why parasite outbreaks can be a sign, not just a cause. They can be a symptom of environmental strain. Now, with that wider lens, you return to the shoreline again. Because the next part of the story is calmer and stranger. It is about the slow helpers of a parasite's plan. Not villains, not minions, just ordinary creatures doing ordinary things. Snails, leeches, insect larvae, tiny drifting crustaceans. They are the quiet middle managers of the pond. They do not know they are moving passengers. They are simply living in a place where everything touches everything. You watch a snail climb, and you can almost feel the physics of its world. The snail is heavier than it looks because water supports it. Its foot grips with mucus, a living adhesive. Its radula scrapes like fine sandpaper, collecting biofilm. And in that biofilm can be parasite eggs, invisible and tough. If you are the egg, this is the moment you have been built for. Not because it is beautiful, because it is likely. Snails graze constantly, and they do it slowly, and they do it everywhere. They are the pond's vacuum cleaners, and every vacuum cleaner eventually picks up something unfortunate. Inside the snail, the parasite becomes a different kind of creature. This is one of the strangest facts about trematodes. Their life stages can look like different animals. An egg hatches into a mericidium, ciliated and searching. Then it becomes a sporocyst, a sac-like stage that absorbs nutrients. Sometimes it becomes a rhedia, a more active larval form with a mouth. Then it produces cercariae, swimming stages with tails. One genome, many bodies. It is like a single story told through different genres. And the snail, meanwhile, keeps doing snail things. It eats, it hides from fish, it moves a few inches and calls it a day, but its internal resources have been redirected. Sometimes the parasite effectively sterilizes the snail. That sounds dramatic, but it is often just an energy shift. Gonads shrink, reproductive effort stops. The snail becomes, in practical terms, a long-term nursery. The parasite benefits most if the snail lives longer. So many trematodes evolve toward keeping their snail host alive, not out of kindness, because a living factory is more productive than a dead one. Now imagine the timing inside that snail. Parasite development speeds up in warm water. It slows in cold water. Cercariae are released in pulses, often when light is right, because light correlates with host activity. Fish and birds have schedules. Parasites learn those schedules without learning anything at all. They are built to respond to cues like temperature and day length. That is what adaptation looks like when you do not have a brain. It looks like chemistry that behaves like a clock. So, in late spring, as sunlight lingers, snails begin to shed cercariae like drifting dust. The water fills with tiny swimmers that cannot afford to waste time. They have limited energy stores. They need a host before their fuel runs out. You drift beside one, and it is almost delicate. A little tail flicking, a small body translucent, carrying enzymes like tools. It bumps into a fish, and it does not ask permission. It penetrates skin or lodges on gills, depending on its design. Then it drops the tail and insists the tail is not needed anymore. The fish becomes the storage unit, and storage is a patient kind of parasitism, not feeding heavily, not causing immediate collapse, just waiting. Now, consider a different helper. Leeches again, but with more detail in their slow method. A leech does not attach like a tick and stay for days, at least not always. Many fish leeches feed, detach, and hide. They prefer brief meals and long digestion. A leech's gut can expand dramatically. It can take in blood and store it, slowly breaking it down over time. That means a leech can feed once and then wait days or weeks. Waiting makes it calm, and it also makes it an excellent carrier. If a blood parasite survives in the leech, it can be transferred to another fish later. The leech becomes a bridge between hosts, not a permanent home, just a vehicle with a syringe built in. Some fish blood parasites are protozoans, related to the ones that infect other vertebrates. They may live in red blood cells or plasma. They can cause anemia, weakness, and reduced fitness. Often the infection is mild in stable conditions, but in stressed fish, it becomes another weight. A weight you never see from the surface. Now think about the leech's behavior again. It finds fish by sensing vibration and chemical cues. A fish swimming near a rock creates a pressure wave. The leech extends, attaches with the sucker, and the mouth parts cut. It releases anticoagulants, sometimes anesthetics, not because it wants to be polite, because a painless bite is a successful bite. The fish may not even notice until the leech is already full. Then it drops off, drifting away, thicker and darker, like a leaf that learned gluttony. It hides in vegetation to digest, and during that digestion, if it is carrying parasites, those parasites also wait. Fresh water is full of organisms that survive by waiting. Parasites simply take waiting and give it a destination. Now, there is another helper that is almost too small to respect. Tiny crustaceans, copepods, and amphipods, the living punctuation of the pond. They are eaten constantly by fish, which makes them ideal intermediate hosts for many worms. Acanthocephalans, the thorny-headed worms, often use amphipods. Inside the amphipod, the parasite forms a cystacanth, a stage designed to be infectious to a predator. Sometimes the infected amphipod behaves differently. It may be more likely to swim into open water, more likely to cling to exposed surfaces, more likely to be eaten. That behavior change can be caused by altered neurotransmitters, or energy depletion, or tissue damage. The exact mechanisms vary, and sometimes it is still debated. But the pattern is consistent enough to be famous. A parasite can turn its intermediate host into a slightly worse decision maker. Not a puppet, just a little less careful. And in a pond, being a little less careful is often fatal. Now let that sink in gently. Parasites do not need to control everything. They only need to tilt odds. A small tilt repeated across thousands of hosts becomes a strong trend. That is how natural selection uses small effects to build big outcomes, quietly, over time, without anyone noticing until you start looking. Now, because you are following the slow helpers, it is worth noticing how many parasite plans depend on edges. The shoreline, the littoral zone, is where hosts overlap. Snails graze there, insect larvae develop there, fish feed there, birds hunt there. Edges are where food webs knot together, and parasites love knots because knots have traffic. You drift along a submerged stem and you see eggs glued to it. They are microscopic, but you imagine them as tiny sealed capsules, waiting. Eggs can be shockingly durable. Some trematode eggs survive cold water for months. Some nematode eggs resist drying for long periods if they are in damp soil. Some eggs can survive passage through a bird gut, which is a sentence you did not ask for, but it matters because it means birds can move parasite eggs between ponds. A bird eats infected fish in one lake. It flies, it rests, it defecates in another lake, and the eggs arrive, ready to start again if the right snails live there. Freshwater bodies are not isolated. They are connected by the movements of animals and by floods and by human activity. Even your boots can move tiny organisms from one pond to another. Mud is an excellent shipping container. Now, stay with the timing theme, because timing is one of the most elegant parts of parasite biology. It is not elegant like a ballet. It is elegant like a factory schedule. Cercariae often emerge from snails at certain times of day. Some species prefer morning, when fish are active near shore. Some prefer afternoon, when water is warmer and larvae swim faster. Some prefer night, when certain hosts are less cautious. The snail responds to light through simple sensory systems. The parasite inside responds to the snail's physiology. So the parasite indirectly responds to light too. A chain of cause and effect that looks like planning. But it is only selection shaping a routine. Now consider seasonal timing. In colder months, parasite development slows. Eggs remain unhatched longer. Larvae remain encysted longer. Transmission drops, not to zero, but to a simmer. Then, spring warmth arrives, and the simmer returns to a boil. Not dramatic boiling, just faster cycles. And faster cycles mean more chances for contact. So when you see parasite outbreaks in nature, they often track temperature. Warm water accelerates many parasite life stages. It also stresses fish by lowering oxygen, so warmth can help parasites twice. It speeds them up and it slows the host down. Now, you drift over a patch of mud and you imagine cysts sleeping there. Some parasite stages insist in sediment, waiting for disturbance. A fish rooting in the mud can stir them up. A storm can resuspend them. A human waiting can release them. The pond floor is not dead. It is a storage layer full of seeds, spores, eggs, cysts, a library of dormancy. And parasites are part of that library. Now, to keep the story grounded, you return again to what a fish experiences, not the whole life cycle diagram, just the fish. A fish swims through a cloud of cercariae and feels almost nothing. A fish grazes plants and swallows a copepod carrying a larva. A fish is bitten by a leech and loses a small sip of blood. None of these are instant tragedies. They are tiny withdrawals, and the fish keeps swimming because a fish cannot pause its life to heal. If it stops moving, it becomes food, so it maintains itself in motion. That is why parasites that cause mild impairment can still be important. A few percent slower, a little less oxygen, a slightly cloudier eye. In a world of predators, small differences matter. Now, speaking of predators, the pond's helpers are not always small. Sometimes the helper is a bird again, returning like a recurring character. Fish-eating birds are excellent final hosts for many trematodes. They have warm guts, constant food intake, and wide ranges. An adult fluke in a bird can produce huge numbers of eggs. Those eggs leave with feces and enter water. Then snails consume them, and the cycle continues. The bird is not cursed. The bird is simply part of the loop. And birds often show few symptoms because the parasites are adapted to them. A final host is where the parasite wants to be. So, it tends not to destroy that host quickly. The intermediate hosts, like snails and fish, often take more damage because the parasite is not trying to settle there forever. It is trying to pass through. Now let the shoreline fade and let the wider lake come into view again. Because once you understand the slow helpers, you can understand the lake's balance more gently. Balance is not an absence of parasites. Balance is an ecosystem where parasite life cycles are diluted by space and diversity, where fish have refuge from low oxygen, where predators keep prey populations from becoming too dense, where plants stabilize shorelines and create habitat for many species, where water quality does not constantly irritate gills and skin. In that kind of lake, parasites are mostly background, and background forces can still shape everything without causing collapse. Now consider one of the simplest ways parasites stabilize populations. They often spread more easily when hosts are common. That is called density-dependent transmission in calm scientific language. In human language, it means crowds share germs. When a fish species becomes abundant, its parasites find hosts easily. Parasite loads rise and host fitness drops slightly. That can slow population growth. It is feedback, not morality. In some cases, parasites help prevent invasive species from dominating, if local parasites can use them. In other cases, invasive species arrive without their normal parasites and they thrive. That is called enemy release. Again, in calm language. In human language, it means moving somewhere new and leaving your problems behind until new problems find you. Freshwater systems are full of these stories. Now consider another balancing role. Parasites can influence food webs by changing behavior. A fish with a cloudy eye may be eaten more often. That means more food for birds and more parasite reproduction. But it also means that fish species might be kept from overpopulating. And when fish populations shift, insect populations shift, algae levels shift, water clarity shifts, everything is connected and parasites tug on connections quietly. They are not the only tug, but they are always there. And because they are always there, ecosystems evolve in their presence. And that constant pressure means you almost never find a fish that is truly alone inside its own skin. You find fish that are coping, you find fish that are budgeting, and you find parasites that are doing what evolution taught them to do, quietly, repeatedly, without any need for drama. If you want a few soft curiosities now, you start by shrinking down again, not to the size of a fish, but to the size of a single parasite stage. You become a larva with one job and a very short fuel tank. You are built out of proteins and stubbornness, and your whole world is water. The first strange thing you notice is how many parasites are basically shapeshifters, not magical ones, practical ones. Your genome is the same, but your body is rewritten for each address. In one host, you are a swimmer, covered in cilia like tiny eyelashes. In another host, you are a sac absorbing nutrients, making copies. In another host, you are a sealed capsule, waiting in muscle like a seed in soil. And in the final host, you become an adult that is mostly reproductive plumbing. That transformation is not a choice. It is chemistry responding to environment, like a lock opening to the right key. Temperature changes the speed. Bile salts change the signals. Enzymes change which genes turn on. You do not need to know where you are. Your cells feel it. The pond writes your itinerary with cues that are older than bones. Now you drift through a patch of moonlit water, and you bump into another oddity: host specificity. Some parasites are shockingly picky, not picky in a spoiled way, picky in a mechanical way. A particular trematode might only develop inside one genus of snail, not because it likes that snail, because its enzymes fit that snail's tissues. A monogenian might only attach well to one fish species, gills, because the hooks match the spacing of gill structures, like a key matching a lock. If you land on the wrong fish, you might not anchor. You might not feed properly. You might get brushed off by mucus and water flow and vanish without anyone noticing. So when you look at a pond as a whole, it is not one parasite problem. It is hundreds of tiny, specialized relationships stacked together. The result looks messy, but the parts are precise. And precision is one way nature hides its cruelty. You drift lower, toward the sediment again, where the water smells like iron and decay. This is where you meet another sleepy fact. Parasite eggs and cysts are often unbelievably durable. They are built like tiny safes, thick shells, sometimes even sticky coatings, so they cling to plants or detritus. A nematode egg can survive cold months tucked in mud. A trematode egg can wait for the right snail to rasp it off a surface. A cyst can sit in fish tissue, quiet as a held breath, until a predator opens the door. This durability is not just toughness, it is timing. It is how you survive a world where most of your offspring never find a host. You do not rely on one perfect chance. You rely on thousands of mediocre chances stretched across a season. That strategy is less heroic than it is mathematical. Now your attention slides to a fish hovering in open water. On its skin, you imagine small scars, tiny dark specks, a couple of pale marks near the fins. And you remember that parasites can leave signatures even when they are gone. A healed leech bite is a small puncture and a little missing scale. A past itch infection can leave subtle gill damage, not obvious, but real. A monogenian infestation can make gill filaments thicker long after the worms are fewer. So when you look at a fish, you are also looking at its history. A timeline written in tissue? Not all of that history is Parasite, of course, but parasites are common authors, and they write in quiet ink. Now you turn toward a detail that is easy to miss, because it is happening inside the fish's head. Some parasites change how fish behave, and the change can be small. Not a zombie fish, just a fish that takes one extra risk. A fish that feeds one extra second in open water. A fish that hesitates just slightly when shadow crosses the surface. That slight hesitation can be the difference between escape and being carried into a heron's gut. Parasites that benefit from predation are not aiming for a theatrical takeover. They aim for a statistical nudge repeated across many hosts. If you want an example that feels almost too on the nose, you look at thorny-headed worms again. Acanthocephalans, those hook-headed passengers. In some systems, infected amphipods become more likely to swim toward light. Light means shallow water. Shallow water means fish. Fish mean transfer. So the parasite's presence becomes a tiny push toward danger. It is not mind control like in movies. It is altered physiology, maybe less energy, maybe changed neurotransmitters, maybe damaged tissues. But the pattern still matters. The pond runs on patterns. Now you shift to another strange fact. Some parasites are basically invisible until you know the trick. Take myxozoans, for example. The name sounds like a mineral, but they are parasites, and some infect fish in painful ways. Many myxozoans were once mistaken for protozoans. Now we know they are highly reduced relatives of cnidarians, distant kin of jellyfish. Yes, you can have a parasite that is, in a deeply awkward sense, a simplified jellyfish cousin. They form spores with little polar capsules like microscopic harpoons. They can infect gills, muscles, even cartilage. Some cause swelling, cysts, or softening of tissue. In certain fish farms, some myxozoans become major problems because crowded fish give spores an easy route. In the wild, many infections remain mild, scattered, diluted by space. But the biology stays strange either way. A jellyfish relative shrunk down into a spore, living inside fish tissue, because evolution loves pruning things until only the useful parts remain. You let that thought drift away, and you settle on a different curiosity. Parasites that change where they live as they grow. Not just which host, but which organ. A larval trematode might enter through gills, then migrate to muscle, then encyst. A nematode larva might pass through the gut wall into the body cavity and coil there. Some larvae end up in the liver or around the gonads or near the swim bladder. The swim bladder is an especially tempting place because it is a buoyancy organ. Even slight damage there can change how a fish holds position in water. A fish that cannot maintain perfect buoyancy drifts oddly. Odd drifting is noticeable to predators. Predators are, unfortunately, excellent at noticing anything that looks slightly wrong. So the parasite does not need to break the fish. It only needs to make the fish a little less smooth, a little less invisible. Now, you follow a fish that is feeding near the bottom. It noses into sediment, taking in small invertebrates. This is where you meet a very practical detail about parasite transmission. Many parasite stages are not floating in open water like magic dust. They are in the places fish naturally put their mouths, in the mud, on plant surfaces, inside small prey. The parasite is not asking the fish to do something unusual. It is attaching itself to normal behavior, the way a burr attaches to fur. That is the deepest trick of parasitism. It uses routines. A fish must eat, a fish must touch the environment, so the parasite just waits at the points where routine meets exposure. Now you move from the individual oddities back to a wider view, and you start noticing how parasites can actually make ecosystems more stable. Not kinder, not gentler, just harder to simplify. If a single fish species becomes too abundant, its specialized parasites have an easy time. They spread faster, they drain more energy, they reduce growth and reproduction, just enough to slow the surge. This does not protect the fish, it protects the pond from monoculture. The pond does not want diversity. The pond simply ends up with diversity when no one species gets to win without paying costs. Parasites are one of those costs. They are like a tax that scales with crowding, and nature loves taxes, even when you do not. Now you look at a different kind of balance, the balance between parasites and pollution. This is where things get quietly uncomfortable, because it connects to your world. When nutrient runoff enters a pond, algae can bloom. The water can look green, thick, almost alive. In daylight, algae can pump oxygen into the water, but at night, algae and bacteria consume oxygen, and when blooms crash, decomposition can strip oxygen hard. Fish then crowd near the surface, or near inflows, or near plant beds. Crowding increases contact. Increased contact helps parasites, especially the ones with free swimming stages. At the same time, low oxygen stresses fish, and stress weakens immune responses. So the pond becomes a better place for parasites, without any parasite changing at all. You do not need a new villain. You only need a slightly worse environment. Now you let the scene shift from wild water to something more contained. An aquarium, a fish tank, is, in many ways, a pond with the safety features removed. The volume is small. The boundaries are glass. The temperature can be stable, which sounds nice until you realize it also stabilizes parasite life cycles. In a natural pond, seasons and space slow transmission. In a tank, transmission can become a straight line. Ick is the famous example again. The parasite feeds under the skin, then drops off to reproduce, then releases hundreds of swimmers. In a pond, many swimmers never find a host. In a tank, hosts are always nearby. Congratulations, probability is no longer on the fish's side. That is why fish keepers quarantine new fish and why treatments often target the free swimming stages. The parasite is vulnerable when it is exposed in the water, not when it is tucked under skin. So the timing of treatment matters. Parasite biology becomes a schedule problem, and schedules are one of the few things humans sometimes handle well when properly motivated by panic. Now drift back to the pond, because the pond is less frantic. Here, everything is diluted, stretched out, softened by space. And that makes it easier to notice a final cluster of strange facts, the ones that feel almost like trivia but have deep logic underneath. One is that many parasites use chemical cues to find hosts. A fish leaves a plume of mucus molecules in water, a faint trail. Snails leave slime trails. Injured tissue leaks amino acids and other compounds. Parasite larvae can respond to these cues like simple machines, swimming toward higher concentrations. They do not smell like you smell. They detect gradients, like following a warm patch in a cold room. That means parasite transmission can increase in areas where fish are stressed and producing more mucus, or where fish are crowded and leaving more trails. Even water flow patterns matter. A slow bay might hold parasite stages longer. A fast stream might carry them away before they find anyone. So, habitat shape becomes part of infection risk. A pond is not just water. It is a set of currents, edges, still pockets, and oxygen layers. Parasites learn that map through survival. Now you slide to a very specific oddity: parasites that live in the eye. It sounds like a horror detail, but it is mostly just strategic placement. Some trematode metacercarii lodge in the lens or vitreous humor. The fish's immune system has limited access there because eyes are delicate. Inflammation in an eye can destroy vision, so immune responses are restrained. That restraint gives the parasite a quieter home. The cost to the fish is reduced vision. The benefit to the parasite is increased. Chance the fish gets eaten. A fish with blurry vision misjudges distance. It fails to spot predators early. In open water, that is the difference between living and becoming a transport vehicle. This is not moral. This is a parasite using the body's own caution against it. Now another oddity: parasites that are basically tiny anchors for bacteria. When a fish louse bites, it leaves a wound. That wound can become a doorway for opportunistic microbes. The parasite is not the only problem. It is also a facilitator. In fish health, this matters a lot. A mild parasite load can become severe when secondary infections pile on. Fungi can colonize damaged skin. Bacteria can invade stressed gills. The fish then spends more energy repairing, more energy fighting, and less energy doing fish things. The pond becomes a place where small injuries ripple into bigger consequences. That is one reason parasites are so tied to water quality. Clean, oxygenated water helps fish heal. Warm, low-oxygen water makes every wound more expensive. Now you let your thoughts land on a final kind of curiosity. Parasites that do not just survive one host's body. They survive metamorphosis. An insect larva becomes an adult, and the parasite persists through the transformation. That means the parasite is surviving a complete rebuild of tissues. Digestive tract reorganized, muscles reshaped, body plan adjusted for flight, and the parasite remains in a protected pocket waiting for the adult insect to be eaten. When you think about it slowly, it is almost impressive. Not admirable, but impressive in the way a lockpick is impressive. It is biology finding a loophole in a process that looks like it should be cleansing. Now, after all those oddities, you return to the calmest, broadest truth. In a normal pond, parasites are not rare. They are managed by the ecosystem. Predators remove heavily infected prey. Diverse habitats spread hosts out. Seasonal cold slows down cycles. Complex food webs dilute transmission pathways, and fish immune systems handle low-level infections as a constant background task. But that management is fragile. If you remove predators, you sometimes keep infected fish alive longer, increasing parasite stages in water. If you simplify shorelines, you change snail and insect communities, shifting parasite routes. If you warm the water, you speed development and stress hosts at once. So, the pond is stable until it is not. And parasites, being opportunists with calendars written in chemistry, are often the first to take advantage. Now you start to notice that this entire story has been about movement. Even the parasites that sit still, the ones anchored to gills, depend on movement. Water movement over gills brings oxygen, and it also brings larvae. Fish movement through habitats brings contact with snails and plants. Bird movement between ponds brings eggs in droppings. Everything is commuting, whether it wants to or not. So you follow one more commute, a particularly common one, because it is so ordinary. A fish eats a small crustacean. Inside the crustacean is a larval stage, folded and waiting. The fish digests the crustacean, but not the parasite. The parasite resists enzymes just long enough to pass into the intestine. It anchors or migrates, depending on its design. Then it waits for the fish to be eaten by something larger. This is why, when you dissect wild fish in ecological studies, you often find larval parasites in the body cavity, coiled nematodes, small cysts, encapsulated larvae near organs. Not because the fish is diseased in a dramatic way, because the fish is part of the pond's transport system. That is what wild bodies are. Transport for other lives, whether they consent or not. Now, to keep this gentle, you focus on a fish that is doing well. It has enough food. It has decent oxygen. It has cover from predators. Its immune system is not overwhelmed. In that fish, parasites are mostly a small tax. A few hook-shaped worms, a few gill-clingy copepods, a couple of cysts under the skin. The fish still grows, it still spawns. It still plays its role in the ecosystem. This is important because parasite stories often sound like everything is sick all the time. In reality, most ecosystems are full of low-grade infections that rarely become crises. The pond is not sterile. It is resilient most of the time. Because it is complicated, now you let your attention drift towards something that is both weird and strangely common. Parasites that alter appearance in a way you can actually see. Black spot is one. White spots from itch are another. But there is also a subtler one. Changes in coloration from chronic stress and irritation. A fish with gill damage may darken in color. A fish with heavy parasite load may look thinner, its head seeming a bit larger relative to the body. In some cases, the belly looks slightly swollen from fluid shifts or heavy internal burdens. These are not guaranteed signs. They are hints. And in the wild, predators read hints well. A heron does not need a diagnosis. It only needs to choose the easiest meal. So parasites, by changing appearance and behavior, can shape who gets eaten. That is one way they quietly influence energy flow in the lake, not by killing everything, but by adjusting the menu. And sometimes the menu changes in a stranger way. because a parasite changes the fish into something that looks like a different kind of meal. You see it in small, quiet distortions. A slightly swollen belly because fluid balance is off. A faint curvature of the spine because muscle has been weakened unevenly. A fish that should hold steady in water but drifts nose up like a tired balloon. Buoyancy is managed by the swim bladder, a gas-filled organ that is always adjusting. When inflammation or infection affects that system, the fish has to work harder to stay level. Working harder means spending calories on posture instead of growth or escape. And if a parasite stage lodges near that organ, the effect can be subtle but constant. In the wild, constant problems are the ones that decide outcomes. Now, let your attention settle on another soft curiosity: the parasites that treat fish as a billboard, not for advertising, but for transmission. Some larvae insist under the skin, and the fish responds by building a capsule. That capsule can become pigmented, creating dark freckles or pale scars that last for months. The parasite is hidden, but the host's response is visible. It is a quiet reminder that immunity is not just fighting, it is also building walls. And walls cost materials. Collagen, immune cells, pigment cells, all of it paid for with food the fish had to find. You can also find parasites that live so gently you would miss them entirely. A few roundworm larvae coiled in the body cavity, doing almost nothing today. They are waiting for the fish to be eaten, but the fish is not cooperating. So the larvae conserve energy like a traveler sleeping in an airport chair. This is one reason parasite strategies seem so calm. A patient parasite can survive long enough to catch a rare opportunity, and the pond is full of rare opportunities, happening all the time. Speaking of rare opportunities, you follow the water upward toward the surface film. The surface is not a clean line. It is a thin, busy skin of oils, pollen, bacteria, and tiny trapped bubbles. Some parasite stages collect here not because they prefer it, but because physics moves them. A calm night can leave a layer of drifting life near the top. Then a breeze arrives and everything mixes again, like someone shuffling cards in slow motion. That mixing matters because it spreads eggs and larvae across space. It is dilution, but it is also distribution. The pond is always balancing those two things, spreading risk and spreading contact. Now, zoom out until the pond becomes one dark shape in the landscape. A pond is never truly alone. A creek connects it in spring floods. A bird connects it with a dirty footstep. A dragonfly connects it by carrying its own history across the air. Parasites move along these connections, quietly expanding their maps. But they only establish when the right hosts already live there. That is the strange comfort in host specificity. Even if an egg arrives, it may have nowhere to go. No compatible snail, no suitable fish, no final host that fits the chemistry. So the pond is protected in a way by how picky the parasites are. Not mercifully picky, just mechanically limited. Now the night deepens and the pond returns to its simplest appearance: a dark surface, a few rings from insects, a slow shifting of plants underwater. Under that calm, Fish continue their ordinary routines. They breathe through gills that never stop moving water. They feed in places where mud holds hidden capsules and eggs. They avoid predators by reading pressure waves and shadows. And they carry small passengers, mostly without knowing it. The passengers are not planning. They are not plotting. They are running old programs written by selection, repeated so many times they feel inevitable. A snail grazes and an egg becomes a larva. A larva swims and a fish becomes storage. A bird eats and a parasite becomes adult. Eggs return and the pond forgets the individual story but keeps the pattern. If you want one last weird fact to keep it gentle and strange, consider that some fish parasites are used as environmental clues. Biologists can sample fish, identify the parasite community, and infer who has been eating whom. Certain parasites only appear if specific snails are present. Others only appear if fish are being eaten by birds regularly. So parasites become a kind of living record of food webs. Not a clean record, not an easy one, but a real one. The pond writes its history in organisms that most creatures would rather not think about, and that is fitting, because nature rarely keeps its records in polite places. Now, you let that knowledge fade back into atmosphere. You return to one fish, passing like a shadow under the moon. Its scales catch light, then lose it again. Its fins make small adjustments, barely visible, just enough to stay balanced. Inside it, life is layered. Cells working, blood moving, gills exchanging gases, muscles storing energy. And tucked into the corners, a few organisms living on the margins, taking their careful share. It is not a moral story. It is a logistical one. A world built from hunger, contact, and timing. The pond looks quiet because the pond is quiet. But quiet does not mean simple. Quiet means the machinery is running smoothly, and somewhere under that surface, a snail is rasping a stem. A leech is folded beneath a rock, full and still. A cyst is waiting in muscle like a seed that does not dream. A bird is sleeping on one leg, digesting the last chapter of someone else's life. The cycles will keep turning without witness. They turned before you were here, and they will turn after you stop paying attention. That is the oddly comforting part. The pond does not require your worry. It does not even notice it. So you can leave the fish to its shadowed routine. You can leave the parasites to their patient math. You can let the lake keep its small secrets, sealed under dark water and soft ripples. And if you listen closely, the last thing you hear is not horror or drama, but repetition.